most promising bio-based plastic for developing suitable packages. Can I have next slide, please? PHA are produced by bacteria from renewable resources, and they are fully biodegradable, but also biocompatible. This is why they can be applied for as packaging, for as bioplastic, but also as biomaterial in the biomedical sector. They have a very wide structural diversity. If I can have this next slide, you can see the chemical structure. In the next slide, please. May I have, yeah, this one? You see that it is a polyester made of hydroxy fatty acid, and the diversity of this polyester lies in the length of the side chain, that is labeled with an R. And we can find then, generally speaking, two main classes. The short chain length PHA, that is, has a side chain up to 2 carbon atoms, like for example, polyhydroxybutyrate, and the medium chain length PHA, like for example, polyhydroxyalkanoate, that contains up to 11 carbon atoms. That's very clear that the chemical composition determines the material properties. For example, PHB is rigid and is mainly crystalline, but the polyhydroxyalkanoate is very elastic and is usually amorphous. So, next slide, please. Can I have the next one? PHA are produced, as I say, by renewable resources, and following the principle of circular economy, they are mainly produced currently from wastes. So, for example, from municipal and commercial wastes after a pretreatment, but also for food industry, like orange juice companies or slaughterhouses, for example, or from sludge in water treatment plants, or from agricultural residues, or even from gas streams, or like syngas coming from pyrolysis of different products, or even for CO2 after the absorption from the atmosphere. In the next slide, you can see a scheme of the process where we can find the upstream, that is the selection of the feedstock, so all the raw materials that I have mentioned. And in the lower part of the slide, you can see the different steps, starting from the selection of the strain that is going to produce the PHA. There are many different bacteria that can produce PHA. You can see that it is accumulated intracellularly in form of these granules, and the content of these granules can be as high as 90% of the cell dry weight. Then the next process is the reactor, the bioprocess itself, the fermentation. And finally, the downstream, that means the extraction of the polymer from the intracellular space of the cells. When we talk about the scaling up an industrial process, in the next slide, you can see that there are different drawbacks. First, the selection of the culture medium, we say the residue, but also it has to be optimized in terms of nutrient limitation, addition, pH, all the conditions. The selection of the strains, also the optimization of the strains, as we will see, that we can redirect the abilities of the strain to produce the PHA that we aim. And finally, the recovery. So the drawbacks came from the fact that it is an intracellular product, and then it is concentrated in the cells. So we need really high cell density cultivation. So the biomass should be at least 100 grams per liter. And then we need a lot of energy to disrupt the cells for the extraction. As I said, we can optimize the strain by cutting-edge technology, means synthetic biology or system biology, and to redirect the flux to the carbon, to the PHA that we want to produce. In the next slide, you will see, I'm not going to enter in details, so I'll show a network, metabolic network, that the bacteria, in this case, Pseudomonas putida, uses to transform, for example, fatty acid into PHA. So you will see in the right part down in the slide, this is the monomer, that is the 3-hydroxyacyl-CoA. That is the monomer that will be polymerized in the polymer, in the PHA. And it is coming from fatty acid. What we do is to modify by knocking in or out gene of these roots that allows the speeding up or slowing down the cycle of the metabolism of this fatty acid to obtain different monomers with shorter or larger chain. You remember I told you that this side chain of the polymer is the one that defines the mechanical properties of the polymer. Then, in my last slide, you will see what are the results of REFUCOAT. In this case, we use substrate coming from food industry, oil substrate, oil residues containing different length of the precursor, the fatty acid, and we produce monomer with different length of the side chain, giving us polymers with different melting point, hydrophobicity, or flexibility. As our coordinator mentioned previously, these different polymers has been used in the project for coating to enhance barrier properties or blending with other polymers. So the take-home message is for scaling up the process, you should use waste resources and to apply different technology for increasing the diversity of the polymer for being implemented in packaging. And that's all. Thank you very much.